zoonotic malaria. Good morning to everyone. We will be speaking both in Portuguese and English, so those who wish, please pick up the headphones for simultaneous translation at the entry. I will be sharing this session with Professor Jorge Snounou, who I'm honored by this possibility, since he's a friend and a collaborator since a long time ago. Please, George. And I will call all the participants. Cristiana Brito. Where is she? Yes, please, come on. Take your seats. Yeah. Érica Braga. And Richard Culleton. And Natalia Figueiredo. Is it? Albuquerque, I'm sorry. As I told, we will be speaking Portuguese and English, so those who wish pick up the simultaneous translation headphones. We have four talks of 20 minutes. And nossa primeira palestrante é Cristiana Brito.
Our first speaker is Cristiana Brito, about whom I'm going to say a few words.
Cristiana Brito é pesquisadora.
Cristiana Brito is a researcher at the René Racho Institute at Fiocruz, Belo Horizonte. She has a PhD and a master in immunobiology at the Federal University of Minas Gerais. She's a biologist at the Catholic University of Minas Gerais, and she's a full researcher at the René Racho Institute. She was the vice director of teaching for the last decade. She had an internship at the York National Primate Research Institute in Atlanta, and she has an important role in scientific dissemination. I would say Cristiana is one of the Brazilian researchers that know the most the situation of malaria and the infection of primates and non-human infection by plasmodiums.
Bom dia a todos.
Good morning, everyone. Thank you, Claudio, for the introduction. I would like to start by thanking the invitation not only of Claudio, but Daniel and Carol as well. It is a huge pleasure to be part of this event. And I would like to thank you for keeping my mental sanity, removing me from administration and management and bringing me here, which is what we like to do. Today I'm going to talk about the molecular basis of zoonotic transmission of Plasmodium from non-human primates. I need the pointer.
Onde que eu vou trabalhar menos? Então, falando de infecção de plasmodium.
Speaking of Plasmodium infection in neotropical primates, we face a paradox, and a very interesting paradox, which is the biggest diversity of primates in the planet. So, in Brazil, we have 171 species of non-human primates divided into 5 families. And we have only 2 species of Plasmodium that naturally infect these non-human primates. These two species, I will speak a little bit more about them, are Plasmodium simium and Plasmodium brasilianum. Plasmodium simium is similar to Plasmodium vivax. It has a very restricted geographic distribution, only in regions of Atlantic forest from the south and southeast of Brazil, and that is capable of infecting only some species of three families of the neotropical primates. Plasmodium brasilianum is similar to Plasmodium malariae, and it has a wide geographic distribution from Central America to the south of Brazil, and it is capable of infecting a large number of different species covering the five families of neotropical primates. In this map, we have a summary of the results of my group in the last 16 years that I have worked with identification of Plasmodium infection in non-human primates, where we already analyzed over 800 samples, finding 108 animals infected. Here we have the different analyses that we carried out throughout this time, drawing our attention to the southeast region, especially Rio de Janeiro and the states of the South, Paraná, Santa Catarina, and Rio Grande do Sul, where we have the Atlantic rainforest and where most of our studies are concentrated. In this image, we see all of the species that my group has worked with, identifying as infected by a species of Plasmodium. In these different colors, we represent the different degrees of threat of extinction of these species, drawing the attention to the last three ones in red, which are the most threatened from the standpoint of preservation. So it's very important to think about the preservation of these species when we're studying the Plasmodium infection. In this chart here, we have a compilation of all of the data already published in literature. Here, only the non-human primate number infected by Plasmodium simium, and in the distribution among different families. So we can see very clearly that there's a preference for the family Atelidae, which is the longest bar of this chart. The Atelidae family, which contains the brown ruler monkey, which is implied as the main reservoir of Plasmodium simium. So in this presentation today, I will try to talk about three aspects. The first one is the invasion of the host. The second one is the potential of transmission, and the third one is the cryptic infection in the brown howler monkey. Regarding host invasion, our first question when we see that chart of distribution of infection in different families is that it's very clear the preference for the Atelidae family, suggesting that there is a difference in susceptibility. So we asked ourselves, is this susceptibility to infection related to the variability in the main molecules involved in the invasion? That is what we studied. Similarly to Plasmodium vivax, the main pathway for invasion is through DBP, interacting with DARK, the Duffy antigen receptor 4 chemokines. So we studied the variability of these two molecules, especially in the regions of interaction of these two molecules to give rise to the invasion. So in this first section here, I am presenting the variability data of DBP of Plasmodium simian. We had a small number of samples and we sequenced only 9 of them. We have represented here only 2 families, the Atelidae and the Sabidae family, as parameters of comparison to a less susceptible family and a more susceptible family. What we can see is that we identified 65 polymorphic sites that gave rise to 8 haplotypes in 469 base pairs. In this haplotypes, we separate the 2 families. I see a high haplotype diversity, and I draw the attention of the central part indicated by the asterisks, which are the regions of polymorphism that differentiate Plasmodium vivax, the sequence of Plasmodium vivax from the sequence of Plasmodium simian that must be involved with the speciation of Plasmodium simian. Here we have represented the non-synonymous polymorphisms distributed between these 2 families. You can see that some of the polymorphisms that are found in the lower part of the chart are common for the 2 families, and the big majority, the vast majority, is specific to one family or another.
Experimento que a gente fez foi
The
mapear
next experiment that we did was to map these polymorphisms. Highlighted here, we have the polymorphisms that are specific to the Atelidae family, which are specific from Plasmodium simium, isolated from the Atelidae family, and in yellow, the specific ones isolated for the Sabidae family. We can see that we have a few differences. And in pink, we have several polymorphisms that are common for these 2 families.
representando também, a gente conseguiu identificar 16
Here we were also able to identify 16 haplotypes,
conseguiram separar as diferentes famílias,
separating the different families with high haplotype diversity and the non-synonymous polymorphisms distributed among the different families. And with the arrow here, I'm drawing attention to only one of these polymorphisms that are exclusive to the Atelidae family. So we need to analyze if this is truly an important site of interaction. Before I go through this, we created a phylogenetic tree using all of the sequences available in databases, and we can see that there is a very clear separation of primates from the old world and the primates from the new world using these dark sequences. And here's a division also in the families, so we can separate them very consistently. Here we mapped the polymorphisms in the 3D structure. We have a focus on this region here, the interaction region. And I'd like to draw your attention to some of the polymorphisms that are present in all of the non-human primates compared to the human sequence, highlighting in blue a polymorphism that is exclusive to the Altidae family, that is more refractory to the infection. We have a very small number of infected animals in this family. Moving on to the second part of my presentation, we asked ourselves if these animals that we call reservoirs, what would be their potential to transmit the infection? All of them have the same infection transmission potential. So To answer this question, we detected transcripts of gametocytes, both for Plasmodium simian and Plasmodium brasilianum. We used only PF25 that we use to amplify the Plasmodium simium that are highly similar, and we designed all the other initiators so that we could access both the targets of feminine gametocytes and masculine gametocytes for both species. For Plasmodium brasilianum, there was nothing described in the literature up to this point. So in this chart, we show the amplification of these targets in 36 samples that we analyzed. And these targets here are only for positive samples. So those that are positive, most of them amplifies PSS25, which is the similar of Plasmodium simium. Some of them also amplified the targets of masculine gametocytes, and we see that there is a preference for the targets of Plasmodium simium compared to Plasmodium brasilianum, and a preference also for the target PVS25 for both species. We used here a fragment that was cloned and gen synthetic so that we can determine the number of copies, and we can see that in most of the samples we are below the quantifiable level of gametocytes. So we identified that there are gametocytes, but we couldn't quantify them.
So we try to quantify this group with PS25 transcriptors, but we had this conversion rate published by COPFIT in 2025. So we had an estimation of the amount of gametocytes in each one of the positive animals, and we don't see very high numbers, but we have some with reasonable numbers of gametocytes. Here we have a summary of our results. We started with 3 samples that were initially negative in our diagnosis, and we saw that 2 amplified for 18S transcript, and 1 had Plasmodium simium gametocyte. And from the samples diagnosed with Plasmodium simians, we had 9 positive only with 18S. They didn't have the detectable gametocyte. 9 had Plasmodium simium, and 1 had Plasmodium simium and Plasmodium brasilianum. The samples that were diagnosed as Plasmodium brasilianum, 7 of them were positive only for 18S, 1 for Plasmodium simium, and 3 for Plasmodium brasilianum. And last, the samples that were known as mixed infections, we had 3 that were positive only for 18S, 3 that were positive only for Plasmodium simium, and 1 that was positive for both species. So what we could see here is that 49% of the samples had detectable gametocytes, a better detection of the Plasmodium simium transcripts, and in the mixed infections, we had a preference of amplification also for Plasmodium simium, as we may see here in this result. So the last part is concerning the cryptic infection. Our group had identified the presence of Plasmodium in bone marrow, liver, and spleens of animals post-mortem analysis. And now we wanted to invest a little more in the identification of the infection in bone marrow. So what we did was to analyze samples of 7 bone marrows of 7 a- animals, post-mortem analysis, and 3 of them had positive for Plasmodium, and one of them we were able to identify transcripts of PSS25 with 7.5 copies per microliter that would correspond to the same factor of correction that was described for Vivax. We used the same. Of course, we don't know if it's similar, but we used, due to genetic proximity, 1.63 gametocytes per microliter. So as conclusions and perspectives, the polymorphisms in DBT and DARK can be associated to infection susceptibility is specific to Atelidae or resistance to infection that are absent in Atelidae. We believe that it's very important to see the combined effect of polymorphisms in the interaction using the tertiary structure. So what is the impact of each one of them in each one of the molecules seen in a joint way? About half of the infected non-human primates has detectable gametocytes. This is showing a great potential for transmission. And it's important to highlight that it also includes other families besides Atelidae. So we consider Atelidae as the main reservoir, but it doesn't mean that the others cannot have a role in transmission. We still need to understand a little more about the amount of gametocytes necessary for the transmission to the vector, since we've seen a very low number of gametocytes, and we observe that the transmission is maintained among non-human primates. So in this minimal amount must be enough to transmit between non-human primates. And last, the cryptic infection in bone marrow that was identified here in naturally infected animals infected with Plasmodium simium, and one of them with detectable gametocytes. So I put as a perspective that we must clarify the impact for diagnosis and especially for the non-human primates translocation, which happens a lot in Brazil. So we must understand and improve the diagnosis to prevent that we have a spread of the transmission, zoonotic transmission within Brazil, and also abroad. We are working with this group that is going to take the brown howler monkey to the north of Argentina. So we must be sure that these animals are not infected and are going to bring zoonotic infection with malaria to this region. So I'd like to thank our whole research group, especially the 3 ones highlighted here, Gabi, Marina, and Loma, who produced these results presented here. And here are all the collaborators who enabled this work to be performed in the last 16 years. Thank you.
Por essa fala absolutamente no tempo, muito informativa.
You were very punctual and very informative. And our next guest is Professor Richard Culliton. I forgot to thank Daniel Barberi and Carolina for the invitation to be here with you today in this very successful event, a high-level event. Thank you so much. I'd like to ask Professor George Nunu to introduce our colleague Richard Culliton.
I'm very happy to introduce Richard Cullerton, a proud Irish Welshman who has opted to go to Scotland to do his degree on zoology. He loved the weather so much he stayed in Scotland for his PhD, which he did with, under the supervision of Richard Carter and David Walliker, both of whom are well-known names and highly original contributors to malaria. After that, I think he got fed up with the wind and he went to Japan in 2005, worked in Osaka, Nagasaki, and then finished in Ehime University, I think in 2020, 6 years ago, and I think you head the department of parasitology over there. His work was recognized by a Masamichi Aikawa Award 2023, and he's one of the rare biologists, malariologists who's interested in more than one species and in more than one molecule, and he knows more about genomics than I do. Next time, choose him. Thank you very much.
Thanks very much, George. That's great. Thanks. I don't know more about genomics than George. I don't know more about anything than George. That's not true. So I'd just like to say thanks to Daniel, Carol, for fantastic organization of the meeting and for inviting me. It's great to be here in Brazil again. It's a wonderful country and beautiful place this is. So I am a little bit jet-lagged and somewhat hungover, so I apologize for that. Also, I'd like to wish good luck to the simultaneous translating team to try and decipher my strange accent, which is a mixture of Welsh and Japanese, as you can probably hear. So I want to talk about zoonotic malaria and how it may be affected by climate change. I thought this was a sort of topical subject to talk about. So, sorry, yeah, I'm in Ehime University in Japan. Obviously, I'm not originally from Ehime. You can tell by my accent, I'm originally from Osaka. It's a different city. So, a zoonosis, as defined by the WHO, is a disease or infection that's naturally transmissible from vertebrate animals to humans. So, and I get that defined before I begin. Of course, you know, host switches occur between different species all the time, between different animals, between different primate species. We only consider a special case if it comes into people. We call this a zoonosis, but host switching is common. So these are a list of the malaria parasites of primates. And just to be pedantic, I put up some terminology at the top there. So sometimes you hear people talking about simian malaria, but humans are simians as well. We're also simians, so we probably shouldn't use that term. So I'll be talking about non-human primate malarias. It's a bit of a mouthful, but those are the parasites which are infective to the other species who aren't us. You can see there's a lot of them. Particularly the ones that are important for today are the ones in the macaques, the old world monkeys here at the bottom. So this is a list of malaria parasite species which are infectious to human beings that we know about at the moment. So when you're writing your introduction paragraph about malaria, it's going to be a long one because there are more than 4 species now which are known to infect people. The ones I've highlighted in red are the zoonotic parasites. So these are the ones that jump from macaques generally into humans, or in the case of simian, of course, as we've just heard, from howler monkeys into people. So there's a Knowles eye is known, right? This is a big problem. We know there's loads of infections of Knowles eye in people. The other ones, and also simian as well, is good characterization of zoonotic potential. The other ones are a little bit sketchier. There's some molecular evidence that they've infected people, but they don't cause, you know, big outbreaks or any kind of clinical problems at the moment, but it's something to keep an eye on. So we currently believe that Falciparum vivax malariae and probably Ovale curtisi and Ovale walliceri became parasites as a result of a host switch event from great apes in Africa, sometime, you know, depending on the species, could be relatively recently. So zoonotic malaria parasites are very, very, very important and very, very dangerous. We know how much disease these species cause, and these were originally zoonotic infections. So this is the areas of the world where you find the parasites today. As we've heard, Piscimeum is down here in the Atlantic forest, Brazilianum in Brazil. And then in Southeast Asia, we've got Knowlesii, Cynomolgi, Inuai, these species in macaques. And in Africa, now these aren't zoonotic as far as we know, but nobody's looked. But the gorillas and apes are infected with a whole host of different Plasmodium species, some of which are related to Falciparum. We don't know if these are infecting people because, you know, nobody's checked. Probably worth a look. So to get to the main subject, I'm going to talk about climate change and the spread of current zoonotic malaria. So just a little bit of background on Plasmodium knowlesii. Its range is restricted currently to this area of Southeast Asia. So the hotspots are in Malaysia and Borneo, Sabah and Sarawak. Naturally, it infects pig-tailed and long-tailed macaques, but causes severe outbreaks. There's a lot of cases in people, like thousands of cases in people in Sabah every year, and it can be lethal. Well, it's rare, but it can be lethal, and it does cause severe disease. At the moment, there's not a great amount of evidence for human-to-human transmission. So anybody that gets knowlesii has been bitten by a mosquito which has previously bitten a macaque, we think. These are case numbers to show you how serious it is. So in Malaysia, all the malaria in Malaysia is now knowlesii. They've got rid of the human cases, now it's all knowlesii. What's really kind of disturbing is that we're seeing increases in cases in Cambodia, Indonesia and Thailand recently. Now, is this a case of we're looking for it now, so we're finding it and it's always been there, or is it coming up? I think that it's probably a combination of the two things, but I do think that the case numbers are actually increasing, and there's various reasons for this, and we can discuss them later. So what is it that constrains the range of knowlesii now, and how might that change? So there are many factors, of course, which constrain a malaria parasite's range. So there's mosquito factors, there's the natural host, there's human behavior, habitat, and there's also the parasite itself, the genetics of the parasite, and also the climate. And I'm going to touch on that a little bit now. So let's consider the mosquitoes. So I know what's coming next, so I'm laughing at this. So the natural vector for knowlesii is the leucocephalus group, which is found in this area, which is where knowlesii is transmitted at the moment, right? We know that, but in the laboratory, you can infect Anopheles maculatus quite easily with knowlesii. It'll go through. Sporozoites come out, they're infectious. So we know that knowlesii could be vectored by vectors which are outside its natural range. I'll carry on talking about that. Also, Anopheles stephensi can be infected with knowlesii. We do that in the lab. Anopheles gambii, we can take to at least the oocyst level, maybe further. Gambii, of course, is the major vector in Africa, so it's worrying. We're not safe in the Americas either. Aztecas in Mexico, Freeborni there in the United States as well, so you're not immune from it there. Quadrimaculatus in Europe, there are competent vectors, and in Japan and parts of East Asia as well. So the competent vectors for knowlesii exist all over the world. What about the primates? What about the non-human primates, the monkeys that they're infected? Well, the natural hosts are these macaques, Presbytes as well, and they're all found in Southeast Asia, around where knowlesii is. But, again, you can actually experimentally infect all of these different species. Some of them are African, some of them are South American. So in parts of South America, we have monkeys which are infectable, and we have the vector, which is also infectable, by knowlesii. So it's something to think about. Of course, there's another host for knowlesii too, which is human beings, and the range of humans is like this. So we're everywhere. So why should it be constrained to Southeast Asia? What's keeping it there? Something to keep an eye on. So what we think, though, is one of the factors is there's very little human-to-human transmission at the moment. But this might change. You don't need to do any special modeling to prove that point, I think. I mean, the fact that we see it restricted to that area shows that there's no human-to-human transmission, in my opinion. But why isn't there? This is an interesting question. So if some of you are clever molecular biologists, you can look into this. So are the gametocytes in human infectious? We see them, we see gametocytes in human infections. We know they're there. Well, are they not infectious? One possibility is the vector isn't biting when the gametocytes are infectious in people. So gametocytes have an infectious window. You know, they're not always infectious. It's possible that the timing of this is different. But might this change? This is the question. You know, parasites evolve and they change. Falciparum didn't used to be a human parasite. It is now. It changed. Could this happen? So parasites adapt. The greater the force of infection, the quicker this adaptation occurs. So the more pressure there is, the more parasites that are out there, the more chance of natural selection doing its work and picking up these mutations. This is a kind of old graph, but it's a scary one, and this shows what's happening to the world at the moment in terms of climate change. Now, this is, of course, man-made climate change is dramatic. The weather's getting hotter. It's caused by people like me flying 40,000 kilometers to give a 20-minute talk, you know. This is something we should think about maybe. But it's getting warmer. How does this affect malaria transmission? Well, it can disrupt vector ranges, it can disrupt the mammalian host ranges, and it can alter transmission rates. I want to talk about this. This looks really scary. Sometimes when I show this, people just get up and leave. But this is the Ross-McDonald equation, and it's useful to know. And it's not as difficult as it looks. R0, of course, is, or R0, if you're American, Patrick, is the number of infections that follow from a single infection. So if I've got malaria and I pass it on to Patrick, but to nobody else, then R0 is 1, right? But if I pass it on to more people, then it goes up. Now, the point is, if you get R0 below 0, then you stop transmission. And all of these factors that I showed, I explained that. The higher R0, basically, the higher R0 is, the greater the force of infections. That's what we're worried about. Now, all of these different letters, they represent different aspects of malaria transmission. This is the number of mosquitoes per person, for example. This is the susceptibility of mosquitoes and humans. And these things are plastic. They can be changed, and they can be changed according to temperature. So if we just look at M, right? So M is the number of mosquitoes per person. So that's very important. So there's 1 person here and 4 mosquitoes. As the temperature goes up, we might expect the mosquito populations to increase. Now you've got more mosquitoes per person, so R0 is going to go up, and the force of infection is greater. Therefore, there's more risk of adaptations occurring. This isn't actually important, but this is an interesting thing to think about. Urbanization, if you're getting lots of people together, what that does is it increases the number of people per mosquito. So R0 goes down when human populations increase because there are less mosquitoes per person because human populations have gone up. Just interesting fact I like to talk about sometimes. Okay, so that's that fact sorted out. This one is a little bit more complicated, but it's important to understand. Mu is the mortality rate of mosquitoes, how fast they die. And T is the incubation time of malaria parasites. Really important, because a mosquito has to bite somebody, but it's not immediately infectious. It takes, I don't know, it depends on the species, but it takes some time before the parasite is then infectious to the next person. And during that time, of course, the mosquito has to survive. So if we take, for example, if this gentleman here is infected with malaria and is bitten by 4 mosquitoes, if the incubation time is 10 days, it's possible that, you know, half the mosquitoes will die during this time. This is why things like indoor residual spraying and killing adult mosquitoes is very important. Because if we can kill the mosquitoes before they become infectious, then R0 goes down, right? Simple, when you think about it. Unfortunately, temperature has a very important role to play in this incubation time. This is a nice graph from Ikimoto's group here. It was published some time ago. But what it shows is the developmental time of the malaria parasite. This is, which one's this? Falciparum, and this is Vivax. According to temperature, so as temperatures increase, the sporogonic development time goes down. So as it gets hotter, the parasite develops more quickly, and this can lead to R0 going going up. It's the same for falciparum and for vivax. So in this case, what's happening is, if we reduce the time by increasing the temperature that the parasite develops, then more mosquitoes survive to become infectious. So R0 is going up because of this. So this is one possible effect of temperature on, or climate change on malaria transmission. And as we get more malaria transmission, again, we get more chance for these things to adapt. So what effect does increased temperature have on the force of infection? So increasing mean temperature and associated rainfall changes will lead to shifts in vector ranges. So we might see the leucocephalus group moving outside its range. This is one possibility. As I just tried to explain, global temperature is also going to shorten the sporogonic development of the parasite in the mosquitoes. And increased malaria transmission will increase the risk of Knowles eye becoming adapted to human beings. So we may see this is very dangerous actually, but if we start to see consistent human-to-human transmission, we're in trouble. With Knowles eye. Knowles eye is a nasty parasite. There's a 24-hour development in the red blood cells, not 48 like falciparum. So parasitemia goes up very quickly. Thanks. Now, we do have some evidence that human-to-human transmission is possible. Jetsmon's group in Thailand showed that they could, if they took Knowles eye out of a patient, put it in a culture flask, allowed mosquitoes to bite on, to feed on it, then they become infected. So it is possible that it could happen. So that's like the bad news, the scary stuff. Now, I just want to touch on this very briefly, because it's not that simple, right? There are other things that can happen. So is it possible that increased temperature can reduce malaria transmission? Well, those of you that were observing this graphic, you've noticed this before, it doesn't continue going up all the way. It does drop off as temperatures get very hot. So when the temperature goes above a certain level, like anybody that's tried to transmit Plasmodium berghei in the lab knows that if the temperature goes above 21, it's not going to go through. So it's possible that we might see, depending on the species, some reduction. Of course, this leads into this factor again. So the susceptibility of mosquitoes is very important. Okay? So, sorry, this is wrong. This is the wrong, this should be here. My fault. Anyway, so the susceptibility of mosquitoes. Not all mosquitoes will become infectious if they bite this person. Of course, some of them won't be. But if temperature goes up, is it possible that we can reduce the number of mosquitoes that are infectious? And I want to talk a little bit about this. This is work that was done by Joel over there. Hi, Joel. Sorry, I'm talking about your work. I don't know why I'm doing it. But this is really nice work, and I just want to put it out there. That so mosquitoes are susceptible to shifts in temperature in ways that you might not imagine. So what Joel and his group did was they reared some mosquitoes in very hot conditions, and they reared them at normal temperatures. And then they took the progeny, and they allowed mosquitoes to feed on, and then these mosquitoes were given a blood mealing of falciparum at a normal temperature. And then you can measure the amount of transmission that goes on. Now, with increasing temperature that the mosquitoes are subjected to, their immune system seems to be boosted, because this is the results from his experiments. At 27 degrees, this is like the control group. The oocyst rate is quite, as you'd expect. But for those mosquitoes whose parents were subjected to increased temperature, the oocyst rates come down. So it may be the fact that, increasing temperatures might reduce transmission. I mean, it's a complicated, multifactorial thing. In summary, zoonotic malaria is an emerging threat, and it's one that we should take seriously. Instability in climate and ecology, of course, can affect the emergence and spread. So I just talked about Knowles eye there, but we might start to see other species becoming problems zoonotically. So it's something that we really should take seriously. And we should do something about it. Now, while we've got the chance, it's emerging, it's getting a bigger problem in Southeast Asia, but it may not stay there forever. We need to do something. I mean, also, people are dying, of course. So to acknowledge, I want to thank Joel for letting me show his data. To Medard, who did the experiments in Joel's lab, Osamu Kaneko, and to, so this is Kamaruddin, who works in Borneo. This is a malaria patient. And this is Cassie, who did some of the thinking for me. That's it. Oh, and that's Dylan Thomas. I should say thanks to him for letting me use his picture. That's it. Thanks very much. Thank you.
Thank you very much, Richard. That's very important because you are leading knowledge eye that is the example of the zoonotic malaria that is the most dangerous, that can lead to the most severe and lethal infections. That's not necessarily the case for cynomolgia and for simian that are not at all severe cases. Let me now call Erika Braga, da UFMG. Erika. Erika é bióloga, mestre e doutora.
Erica is a biologist, master, and PhD at the Federal University of Minas Gerais. She had a doctorate in London School and the Museum of Natural History in Paris, where she worked with an extraordinary group of parasitologists, Chabot, Irene Landau, François Milgan, Odile Ban, an extraordinary group of parasitologists. She's the full professor of the parasitology department and leader of the interdisciplinary group study in malaria. She focuses her research on the immune response to malaria, especially in infections by Plasmodium vivax and aviary malaria that she's going to talk to us about.
Bem, em primeiro lugar, bom dia a
First
todos.
of all, good morning, everyone. I would like to thank the organizing committee for the invitation, and I would like to say that I took this challenge of speaking about avian malaria to a team that works basically with human malaria. So I'm going to show you how we can use the model of avian malaria and the correlated parasites to understand the effects of the anthropic actions, modulating the transmission dynamics of vector-transmitted diseases. To start, I think it is important for us to understand that the human being has changed the environment from thousands of years, and more recently in the last few centuries, especially after the Industrial Revolution, when capitalism started being our current economic model. The consequences of these changes are disastrous, and we observe the emergence of certain diseases, the appearance of new diseases, the extinction of species, and the loss of biodiversity. Today, in the planet, you practically don't find a place where we have no effects of the actions of human beings. And practically half of the planet Earth, we have areas that have from moderate to high anthropic pressure. This causes sensitive effects in the ecosystems. Thus, what we have seen is that we have used a few predictive models aiming at understanding the effects of anthropic actions to be able to propose measures that could minimize the damage caused by these actions that we are imposing to the planet. So we have used a model called avian hemisporidians, and I'm going to try to explain to you why these models are interesting to analyze these anthropic effects. As a parasitologist, as Claudio said, participating in groups that still work on classic parasitology, find it important to understand who are these hemisporidians. These are not only Plasmodium genera parasites, but we have other genera like Hemoproteus and Leucocytozoom, They are diverse hemosporidians because we have several subgenera. This is proposed based on biological characteristics of these parasites associated to the vertebrate hosts, but especially in vector insects. The plasmodiums that infect birds, they belong to five genera. These subgenera have very remarkable biological differences. The avian plasmodiums, they affect a huge amount of birds in different ways and differently than the human and mammals plasmodiums. They are transmitted by vectors, vector mosquitoes of the Culicines family. So they have a different biology. In the Hemoproteus genera, we have two subgenera, Parahemoproteus, that infect a lot of birds as well, transmitted by midges. This is a different transmission here too. And the subgenera Hemoproteus that infect primarily uh, pigeons. I call pigeons flying rats. So they are transmitted by louse flies. And these louse flies, they remain with the host pretty much throughout the whole development. They are in between the feathers of these birds. And we have the leucocytozoon, whose subgenera is only leucocytozoon that infects different types of birds that are transmitted by black flies. These are parasites that have a lot of biological alterations, and we hope that this could be a model when we work with climate change because the vectors are especially different and sensitive to these changes. So our hypothesis was that the anthropogenic and the climate factors modulate the dynamics of transmission of these parasites. And in order to analyze that, we use climate variables. We'll work with three of them: temperature, seasonality, and precipitation. And we also work with variables that are anthropogenic to measure the degree of human influence on these ecosystems. And we worked with two important variables: the human influence index, also called human footprint index, and the normalized difference vegetation index, NDVI. These anthropogenic variables were measured through remote sensing and computational analysis. And we analyzed this in a period of 19 years, as I'm going to show to you now. What is this human footprint index or human influence index? It is a measurement that takes into consideration 8 factors that represent this human pressure on ecosystems. So, built environments, population density, nighttime lights, croplands, pasture lands, roads, railways, and navigable waterways. They are part of the human actions on environment. This index was proposed in 2020 by Sanderson, and then it was revised by Venter in 2016, and it takes into consideration these impacts of the human activities that goes from 0 to 50. The areas with the human footprint index below 4 are considered intact areas where you can have a preservation of the ecological relationships in these ecosystems. When you have indexes from 4 to 7, frequently these are areas that are already subject to agriculture and livestock with a significant alteration. And those areas that are considered having an HII above 12 are highly impacted areas. What we can extract from this index is this online platform on the right-hand side, WCSHumanFootprint.org, and this platform gives us satellite images from 2000 to 2020. On the page, you can see South America, and the yellow areas have higher HIIs. The green areas have lower human influence indexes. They use satellites. The images are obtained by satellites that go from 2000 to 2015 with first generation satellites, and from 2015 onwards, they use second generation satellites, improving these images with time. Another measure that we use to assess the anthropic action is NDVI. NDVI is very much used in agriculture, and it measures the health of the vegetation and vegetation coverage. The healthy plant, for it to go through photosynthesis, it absorbs the visible light, frequently red light, and it will reflect infrared, which is not visible. So this index takes into consideration this relationship where we have absorption of red light and reflection of infrared light, which is going to increase the levels of NDVI, showing that you have a lot of chlorophyll per unit of biomass, and consequently you have plants that are healthier and a healthier vegetation.
This measure that we use, NDVI, is obtained from satellite images, but using this online platform called Google Earth Engine. So these are the data. We have this large database in our lab. Since 2020, since 2001, we've been obtaining sequence of the parasites, all the parasites that I've shown in the beginning. And the sequences that are showing the infections are part of this database. And we also use data from literature about prevalence of hemospherite infection. And we work with around 2,600 birds. From them, you have 194 species, distinct species, a great variety, representing 16 orders of birds. We also analyzed all the database, and we observed that in Latin America we have 249 analysis sites where we collected the samples, and the birds occupy especially the tropical forest biome and subtropical forest biome, either humid or dry. So each community we were going to study should have at least 10 birds. So these communities should have at least 3 distinct species of birds to guarantee heterogeneity. And with that, we defined 58 communities. And these communities are basically in Brazil and Peru, and with a single community in Argentina and another one in Ecuador. So most of the data we have are coming from Brazil and Peru. So we worked to define the prevalence, and we used the amplification of this mitochondrial gene. This is the standard for the avian plasmodic studies, and our variable then is the infection itself, the response variable. And what we have is 24.6% of total infection among the 200 uh, among the 2,600 birds we evaluated. Considering the genre and subgenre, the Hemoproteus is the more prevalent, and and then Hemoproteus that we have we have flies adhered to the animal bodies, and then with Plasmodium and Leucocytoson with a very low prevalence. And this is expected. Leucocytoson is a species that has a very restricted distribution in Latin America, especially in the Andes countries, and also with high altitudes that is related probably to the biology of the vector. And leukocytosis was not used in the models because of the low prevalence in an individual way. It was only considered in the main model. And something else, since the distribution for the other genera was not very different, the biome was used as a random variable and not as an explanatory variable. So this is the model for all the genera evaluated together. And what we notice here are the climate variables and the anthropogenic ones. So here, for instance, we have the probability of infection at the y-axis, and here the yearly temperature. And what we notice is that in the case of temperature, we have this unimodal standard showing that the temperature between 10 and 15 has a greater probability of infection by hemosporides. And we observe here the seasonality of temperature, and this is an important piece of data because the temperature seasonality is going to indicate thermal variations. And we notice that in this unimodal pattern that minimum temperature variations are capable to make this probability negative, uh, the hemospheride infection. And concerning annual precipitation, we have this linear uh, relationship, and with the increase of precipitation, we have a lower probability of an infection. Now, concerning the anthropogenic parameters, the human footprint index with the influence index, which is unimodal standard, that is showing that from 10 to 15 that we have impacted areas enabling this greater infection. Concerning the vegetable coverage, we have this linear relation showing that areas with more vegetations, we have a greater possibility of infection. So they seem to be opposite data, but we normally think that everything that is green is good. But what we see here is that this green is that the agriculture is good. So in reality, these are crops showing that this is healthy, not necessarily that is a preserved area. So these are not data that are contradictory. So when we evaluate separately for the different genera, what we see here is concerning Plasmodium, and it's transmitted by Corycineus. So this precipitation between 1500 and 2000 is ideal to promote this infection. And it's different for Paramoproteus because we have this total prevalence. So this Hemoproteus has a greater infection. It's modulating the module when I use all the parasites jointly. So it's interesting to observe the temperature seasonality. We see the same general pattern showing that for both parasites, small variations are able to change the dynamics between the parasite and the host. Concern the impact, we weren't able to see this in association with NVDI. So there's no change concerning vegetable coverage, but we have that for the human footprint, showing how much these pressures are modulating the transmission of these two genera. But we see a clear difference here because for Plasmodium, the peak of this infection probability is precisely between 10 and 15, between 5 and 10. And for parahemoproteins, we have this greater threshold, showing that the parahemoproteins, this is more adaptable than the Plasmodium. But we are clearly seeing that these changes are modulating and increasing the infection probability. So human actions are being reflected in the infection probability. And an important piece of data is that if we analyze Colicinius, and here we have um, anthropicized areas that we know we have a lot of transmissions of Aedes aegypti with the dengue fever virus. So this is corroborating these results. And when we observe for hemoproteus, we also have the vector of Urupushi fever that is being found in degraded areas around forest areas and rural areas. So this is lighting an alert showing that this is an interesting model. Just to wrap up, concerning the hemoproteus and the parahemoproteus, that the parasite is close to the host and the vector, the columbiforms are the only model that we were able to correlate the time evaluated. So we take from 2009 to 2015, and we have an increase of the prevalence, therefore, in the infection probability. And this is reflecting the pastures that are now an ecosystem that is very much used by the birds and is maintaining the 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 breeding sites with humid uh, organic matter. So this is a model that is very much used for the loss of biodiversity, because what happens is that the columbiforms in these regions, you end up selecting species that are synanthropic and that are dominating the ecosystem there, keeping the parasite. So these are our conclusions. The human footprint index is a strong predictor of Hemosporidian prevalence with a consistent unimodal pattern. The Hemosporidian infection peaks at intermediate high HII, indicating that moderately disturbed environments maximize transmission. The Plasmodium prevalence peaks at moderate HII, likely due to optimal mosquito abundance and host vector parasite interactions. So we can have several vectors in these conditions. In the case of Parahemoproteus, it's more adapted to the changes than the Plasmodium, and we don't have any association. So to end, because Claudio is nervous, the main conclusion I have for you is that avian malaria and related Hemosporidian parasites serve as a useful model for studying the transmission dynamics of various vector-borne diseases, and that avian malaria matters. So I wanted to thank, and I'm very moved because this is a work developed by women. Daniela De Angeli is at Bangor University, Wales. She received Liz Marques Vieira and with the program of CAPES, She sent me the data last week. So now we are going to include more than 11,000 birds in our next analysis. And I wanted to make a joke. These are satellite data. I wanted you to read what's being shown here with the letters. Even malaria matters. Even malaria matters. That's it. I hope you liked it. Thank you, Erika, for your informative presentation and training presentation, showing how important this avian malaria model is. And now we're going to have Natalia de Albuquerque, who is a doctor and a doctor in genetic and molecular biology. And during her doctoral studies, we had this internship in computational biology, focusing in comparative genomics. She is a postdoctoral student at USP, where she applies population genomic tools. Thank you. Thank you.
So good morning, everyone. I want to thank Carol and Danielle for inviting me to speak. And I'll talk about Plasmodium simium, birth and evolution of a zoonotic malaria parasite species. And first, I want to talk a brief history of how simium was discovered. So it was first reported in 1951 as a malaria parasite that was infecting howler monkeys other than Plasmodium brasilianum. Plasmodium brasilianum was already known. And they named Plasmodium simium. In 1966, they reported the first human infected with Plasmodium simium, and also suggested the Anopheles cruzi, the subgenus Cartidia, was responsible for the transmission of this parasite because it has a vertical behavior. They can bite monkeys at the trees and humans at the ground level. So they warned for the zoonotic potential of Plasmodium simium. And they were right, because in 2017, the group of Professor Claudio reported an outbreak in Rio de Janeiro between 2015-16, and they detected 28 samples of Plasmodium simium infecting humans. And they diagnosed simium by microscopy by two SNPs in the mitochondrial genome that were thought to be exclusive to Plasmodium simium. And these infections in the Atlantic forest was usually attributed to Plasmodium vivax. The first nuclear genomes were sequenced in 2021, the two groups of Professor Claudio and the MOA lab of Professor Marcelo. And they could answer important questions about this parasite. One important thing is that Plasmodium simium is highly similar genome-wide to Plasmodium vivax, but they have important differences. One important thing is the deletion in reticulocyte binding protein 2A and the Duffy binding protein 1. And also, they showed the order that it happens, the host shift. First, a subpopulation of Plasmodium vivax jumped to monkeys in anthroponosis, and then back to humans as a zoonosis. And they showed a larger amount of the genetic diversity in Plasmodium vivax in relation to Plasmodium simium, and also Plasmodium simium of monkeys with this larger amount of diversity compared to strains isolated from humans. So this is the order. And also, they show the deletion in RBP2A was detected only in Plasmodium simium, both from monkeys and humans. So this is a useful marker to distinguish vivax from simian. They also suggested that Plasmodium vivax arrived from Europe at the time of the colonization, and then they adapt to this new environment and the new vectors they encounter in South America. And this is the hypothesis I work in this paper. And also, I wanted to address these knowledge gaps so far. Is Plasmodium simian an independent taxonomic unit, or is there gene flow between simian and vivax? Is Plasmodium simian closer to Plasmodium vivax from South America or Mexico, as suggested by these previous studies? And is there genetic differentiation between Plasmodium simian from monkeys and humans? So first, I want to show you the Plasmodium simian originated from a recent host shift. And here we compare 3,060, more than 600 orthologous gene pairs between simian and vivax. We estimate the diversity, the nucleotide diversity, pi, and the log ratio. First, the null distribution here, centered on 0, is what we expect under identical nucleotide diversity between these species. And since the distribution is shifted to the left, it shows us that there is less diversity in Plasmodium simian than in vivax. We also show this with this method, the pairwise sequentially Markovian coalescent analysis. This method uses the coalescent theory because the rate of coalescence can tell us about the effective population size, because coalescence events are more likely to occur when the population is small. And here in this graph, here it's older time, going backwards in time, and here is more recent. And the first thing that we can notice is the different historical trends of effective population size for Plasmodium simian and vivax populations. Plasmodium vivax has a much deeper genealogy in comparison to Plasmodium simian in red. We also can see this drop of effective population size through time, and this sudden drop in more recent times. This is suggestive of a sharp population expansion following a bottleneck that could have happened when Plasmodium vivax arrived from Europe. And Plasmodium vivax show consistently a small population, and also the small population size is consistent with a split from a common ancestor with Plasmodium vivax. Here, I want to show you the affinities of Plasmodium simian. We included in the data set of simian 12 potentially Plasmodium simian human-derived isolates from São Paulo. And in the PCA, we can see that all these samples cluster here and is closer to Latin America as we expect. But zooming in only in Latin America samples, Plasmodium simian samples cluster closer to Brazil Plasmodium vivax population. And we also did F4 statistics using Plasmodium simian populations, comparing with Plasmodium vivax populations of Latin America, with Mexico population center in 0, and we see that Plasmodium simians share more alleles with Plasmodium vivax from Brazil than with the rest of Latin America. And to confirm that these samples from São Paulo are indeed simian and not vivax, we compare only the samples from São Paulo with simian from Rio and Espírito Santo, and the other samples of vivax from Brazil, and they share more alleles with Plasmodium simian. Next, we search for genomic regions under positive selection. We use this method of regions identical by descent shared by the population. Because positive selection is expected to increase these regions at the target locus and neighboring sites. So we see that Plasmodium simian has peaks in chromosome 3, chromosome 9, 12, and 14. And what is interesting in these regions? We could identify in chromosome 3 the AP2 domain transcription factor that is involving oocyst maturation in the vector, the 6-cysteine protein that is involving sporozoite invasion of hepatocytes, and in chromosome 9, the apical membrane antigen 1, that is an important vaccine antigen candidate. And it's important to notice that this detects recent events of positive selection because with time, these long haplotypes are break in the recombination. So this is in the last 25 generations back. In here, I'm going to show the gene flow in Plasmodium simian population. This is a network plot connecting samples that have their genomes identical by this, their genomes more than 25% identical by descent. So they have roughly a half-sibling relationship. And we see that samples from Rio and São Paulo share these identical by descent, which shows recent gene flow between states. And also here, the gene flow between simian from humans and from monkeys. And also, we can see that simian from humans and monkeys also share more than 25% of genome in IBD. So there is gene flow. We also sequenced old samples from Sado Valley, Portugal, from the 1950s. This region is very interesting because it was one of the last regions in Europe to eradicate Plasmodium vivax. These samples were given to us from the Health Museum by Helena de Andrade. We extract the DNA from these blood slides, and we used two different methods of DNA enrichment, selective whole genome amplification that we sent to University of Maryland with the help of Professor Joanna, and hybridization capture that we sent to Max Planck with the help of Megan Mitchell. And one of the samples, the sample 3 that we call Portugal 3, we could recover a lot of reads, and we have it is our most, the highest quality sample we have, that it has 15,000 SNPs, so it's the best quality of historical samples so far. And then we saw something very interesting about Portugal 3. When we did Smart PCA, that is a technique to calculate the PCs using the more modern and most with the highest quality samples, and the older samples are projected in this PCA space. And Portugal 3 clustered together with Plasmodium simium, which is very unexpected. And we also did F4 to test for the allele sharing between Portugal 3 and Plasmodium simium with the rest of Plasmodium vivax modern populations. And they share much more alleles with Plasmodium simium than with the rest of Plasmodium vivax populations. And about the reticulocyte binding protein 2A that I told you that is characteristics of simium, this protein is missing 44% in the Plasmodium simium. So it's like it disrupts the protein. So there's two possibilities, or they are still working, but it's very less efficiently, or Plasmodium simium uses another protein to do this ligation. And what about Portugal 3? We found a deletion in the exact same place, and the neighboring regions have a very good coverage of reads. So we can say that Portugal 3 have this deletion that is characteristics of Plasmodium simium. But they don't have these SNPs in the mitochondrial genome that was the initial way to identify Plasmodium simium. And unfortunately, we don't have enough coverage in the BP1, so we can't see if there's a deletion there or not. Another genomic signature of adaptation is the PV47. That is a protein involved in parasite survival in the vector. It's used to evade the mosquito immune system. And this study showed that there's different haplotypes that predominate in old and the new world. And Plasmodium simium is closer to Brazil and other Latin America samples. And they have this difference here. And the P3 have the same haplotype as Plasmodium simium that is different from the other Latin America samples. And these non-synonymous mutations are associated with vector compatibility. So it's suggested that this is because of the adaptation to the new vectors in South America that these parasites encounter. So I had two hypotheses for the sample, what simium are doing in Portugal. So maybe Portugal 3 represents an intermediate lineage between simium and vivax that circulated in Europe before the eradication. It was introduced in Brazil. Or Portugal 3 is a lineage of simium lacking these mitochondrial SNPs that was imported into Europe from the Atlantic forest of Brazil. So in summary, we see a genetic similarity to now extinct European lines of Plasmodium vivax with the Brazilian and Latin America Plasmodium vivax. There's this unique deletion in RABP2A and PV47 SNPs in Plasmodium simium that can distinguish from vivax. We saw gene flow between simium samples from Rio and São Paulo, and also from humans and monkeys, and this presence of Plasmodium simium in Portugal in 1952. So I want to thank you, my supervisor, Marcelo, Joanna, and all my lab mates and friends. Thank you very much for your attention.
Muito obrigado, Natália. A organização me pediu que
Thank you,
reduzisse
Natalia.
a discussão para 10 minutos.
The organizers asked me to reduce the time of discussion to 10 minutes, so I would like to ask you to ask short questions and to ask the speakers to have short answers as well.
Discussion time to 10 minutes, so I would ask the colleagues to make short questions and the speakers to give short answers.
I think all the talks were really amazing, really enjoyed all of them, but I have just two questions. One is for Cristiano, which is that 3 out of 3 of the monkeys had cryptic infection that you tested, but I mean, that must have been selected in some way that you suspected they would have it, or are you concerned that there's a lot more cryptic infections in both monkeys and maybe in humans as well than is recognized? So I'm wondering how the selection of that is. And then for Natalia, it is, what about CSP repeats, and is there diversity in the CSP repeats in the simian, or is it a single one of the variants? But I have a lot more questions, but we're told not to ask a lot.
So for the critical infections, we have only 7 samples. So these samples, it's post-mortem samples. So we have a difficult to conserve the samples. So because of that, we don't have a lot of different, a higher number. So I think we can, you can try to amplify the orders, because we have 7 samples already studied. But I think the parasitemias is very low in circulation, so we don't know if some of the parasites are in the critical sites. Because of that, it's so difficult to find in the circulation. But I think we have very few samples to have a conclusion about that.
Okay. And the CSP?
And I haven't checked yet, but I will.
Okay, thanks.
Cris, eu acho que toda vez que você dá essa palestra, eu te faço essa pergunta. E eu sempre esqueço a sua resposta, então eu vou fazer de novo.
Chris, I think whenever you give this lecture, I ask this question and I forget the answer, so I'm going to ask again. You detected 3 Vivecs in non-human primates, 3 detections of Vivecs, right? On the marrow? On the bone marrow? No, I wanted to ask if in these samples, if you checked the same thing that you checked for simian, if there is Vivecs gametocytes, transcripts of gametocytes. I don't know, because we're using the same initiator. So we detected transcripts of gametocytes using PVS25 mainly, which is from Vivecs. So we don't know how to differentiate it from what we did. Couldn't differentiate if it was Vivecs or simian. So on these samples, it could be Vivecs as well. But in our diagnoses, we do the differential diagnosis of Vivecs, from Vivecs to simian, using one of, as Natalia said, one of the mitochondrial ones we use it for differentiation. So those were simian. We did the differential diagnosis, but the gametocyte specifically was not analyzed.
Hello, for Chris. So I really enjoyed the talk, and also Richard's talk, and trying to connect both. So you mentioned about the transmission potential of these these infections, and you have very low gametocytemia levels, and you were wondering if they are transmissible or not. As Richard mentioned, there are a lot of components that it's connected with transmission and everything. So if you have very low gametocytemia levels, and for example, if the monkeys or the other non-human primates stay infected for a very long time, and you have a lot of mosquitoes, even though if you have very low transmission levels from these organisms to mosquitoes, it can sustain some transmission. So my question is, do you have an idea for how long these non-human primates can remain infected and the prevalence of infection in the wild? Thank you very much for the presentations.
So I don't have the idea of how long, but they will never be treated. So we analyze, for example, samples from Rio de Janeiro that we follow for more than 10 years, and we have infection in the many different times in that interval. And we did some years ago genotyping using microsatellites of these in different times, and you can see that the genotype was the same in, for example, in 2011 and 2020. So maybe it's the same, is a chronic infection. But for simian, for Brasiliano, we don't know if they have the spontaneous cure, but we have some data as well that we can identify infection in different periods in the same monkey. That data is for the same monkey that we took samples in the captive. We took samples in different times. So we expect that for both the infections are chronic. And in sometimes the genotype is the same along the time.
Thank you, Cristiano. George, we have a last comment, please.
A quick comment for Natalia's work, and it's very, very interesting. The third possibility is that P. simian was introduced to Portugal from Brazil. Okay? Now, I think people, do you call it Portugal free? I don't need to remind this audience that the Portuguese and the Spanish, way before the British and the others, had moved all around the globe and came back, probably bringing parasites from all varieties of countries. And there's nothing to say that the European vivax is a European vivax, the samples that you've had, especially in the 1950s, because it can't well be that the simian was introduced to Portugal, and Dr. there is a Brazilian parasite in Portugal. It's equally plausible. Okay, that's it.
Okay. So we have to thank all the speakers.
Claudio, posso fazer uma última
Daniel,
pergunta?
você quer dar um aviso ou você vai fazer alguma pergunta?
May I ask a last question? You told me to go fast.
Go ahead. Natalia, he has a question.
Just following the question from the question or comment from George. So if this parasite was circulating in Portugal, wherever coming from Brazil or from anywhere else where it was circulating, why do you think it disappeared? I mean, we know why it disappeared in Portugal, but why it disappeared in Brazil from human transmission? I mean, why would we don't find it anymore in humans?
No.
Well, because they're
You're
not
talking about
that
simian?
Yeah. you can
because
make it.
there are not that many infections of simian in humans today, I don't know how many monkeys were infected with simian.
But then you're calling it a simian. I mean, if it's
Well,
an
you call it
intermediate
simian.
parasite that was circulating in humans in Portugal.
We agree that simian is an adapted P. vivax parasite. Is a P. vivax parasite that adapted to the local primates here. Unless you said that there was P. vivax before the Europeans arrived here, which might be possibilities, unlikely. So it's to all extent, and 500 years is really not a very long time in evolutionary terms. It's adapted a little bit, it's lost a few genes, it added a few more, it moved from one to the other. I don't want to put an arrow from man to simian, to monkey, and then many years later it comes to human. Maybe this has been circulating amongst Brazilian, the whole of Latin America. I mean, there were cases, as you said, Mexican, there were cases of P. vivax looked in other countries in Latin America that could have been vivax gone into monkeys, and then monkeys back into vivax. I was telling Patrick Duffy, in 1931, they described a vivax-like parasite in Panama, from Panamanian animals. So they're susceptible to vivax. Why shouldn't they have transmitted it back to humans?
No, the point is not that. The point is that she finds a parasite that has a deletion in RPP and doesn't have the allele, mitochondrial allele, which is simian, right? So that would be an intermediate parasite.
Why don't you find it
so
here?
It came and it disappeared from humans because we don't find this parasite anymore in humans. Why it disappeared? You see?
Shit happens, as they say.
This is a subject for the lunch discussion. Ok. A que horas eles têm que voltar, Daniel? Caroline, que horas eles têm que voltar?
At what time should we return? At 2 p.m.? Even if you're late, there's no tolerance? No tolerance, okay.
O almoço é aqui no salão do lado.
We're going to have lunch in the next room. Thank you if you stayed up until now.